We're going to talk for a few minutes about the Streptococci, and in this first video we're just going to talk about the basics. Who they are, what they do in real basic terms, and then in the subsequent videos we'll talk in more detail about group A strep, group B strep, pneumococci, oral streptococci, and then finally the enterococci, close relatives of the streptococci. So as the name should imply, these are gram-positive cocci. They can form streptococcal chains. They're often found as diplococci, relatively small, anywhere from half a micron to 2 microns in diameter. And typical of cocci, they're non-motile, no flagella, no swimming, and they don't form endospores. They are all fermentative in their metabolism. There's no respiration going on, so therefore no Krebs cycle, no electron transport chains. Some of them are obligate anaerobes, meaning they can only ferment and therefore metabolize and survive in the absence of oxygen. Others are aerotolerant, meaning that they will still ferment in the presence of oxygen, and the oxygen doesn't suppress them in any way. The catalase test that you see down there in the last bullet is a very handy, quick, easy test to do, and it helps to distinguish the streptococci from the staphylococci very rapidly. And so it's valuable to know that the streptococci are in fact catalase negative. If we haven't already done it by now, you will learn the catalase test in lab. All right, streptococci can be found all over the human body, but they are most often found in the pharynx and in the mouth itself. In fact, most of what we find in the oral pharynx is in fact various species of Streptococcus. They can be found in the conjunctiva of the eyes, sometimes in the nose. They can be found transiently on the skin or in the lower intestinal tract as we swallow them and they pass through. More often in the lower colon we find the enterococci. They're enteric cousins. Maybe enteric isn't the right word, but they're gut cousins. And then on occasion they can be found in the female urogenital tract as well. But primarily when we think streptococci in the human body, think oropharynx. Now you've heard terms like group A strep and group B strep. These are based on a classification system that was developed by a woman named Lansfield. And what she designed was a very rapid way to differentiate the different streptococci based on surface carbohydrates, so capsule carbohydrates. Now carbs typically aren't great antigens, but in this case we can get a pretty good antigenic response against fluorescent antibodies, and they can essentially light up if a particular antigen, a particular carbohydrate, is present. So when we talk about group A streptococci, we're talking about various strains within the species Streptococcus pyogenes. That's group A strep. Group B strep would be various strains within the species Streptococcus agalactiae. And then there are a couple other groups that don't seem to have as much importance in human health. And then there are some that we call non-Lansfield streptococci, meaning that this Lansfield system of using antibodies against carbohydrates on the surface is not particularly effective at distinguishing them. But these are important either pathogens or opportunists. Streptococcus pneumoniae, more often called the pneumococcus, and then the various oral streptococci. And in older publications, you'll see the term viridans streptococci. So if you see that, recognize we're talking about oral streptococci. And in particular, the species mutans, sabrinus, and salivarius, all of which can be involved in causing cavities and various forms of gum disease. Mutans seems to be the most guilty in that process. And then finally, it's important to know that many of these carry hemolysins, right? Cytolytic toxins. You've learned about those exotoxins that bust apart cells. This is a photograph of a blood agar plate, basically LB agar with red blood cells in it. And someone was clever and took a loopful of 3 different types of streptococci and made an alpha, beta, and a gamma. Those Greek letters are how we classify them. I don't expect you to memorize which are alpha, which are beta, which are gamma, but I want you to understand that there are different ways that hemolysins can express themselves on a blood agar plate. You see, for example, alpha hemolysis has kind of a greenish twinge to it, whereas beta hemolysis goes completely clear and you just have the color of the LB agar shining through. And then remember that gamma hemolysis is some smart guy's attempt to make non-hemolytic sound cool. Gamma hemolytic means non-hemolytic, so keep that in mind. We see that some of them can be distinguished quite nicely. For example, we can distinguish Streptococcus pyogenes, group A strep, from the pneumococcus based on its hemolytic pattern on blood agar, because the pneumococcus 99% of the time is going to be alpha hemolytic, and Strep pyogenes is always going to be beta hemolytic. Others, like group B strep, Strep agalactiae, not so easily categorized because there are a variety of hemolysins in circulation, and even some that aren't carrying any hemolysins. And to some degree, that's true with the viridans strep. More often than not, they're alpha or non-hemolytic, and every now and then you'll find a beta hemolytic oral streptococcus as well. So again, don't memorize this image here, but get an understanding of the fact that there are different types of hemolytic patterns, different types of cytolytic toxins, and the different groups of Streptococci can often be distinguished based on their patterns of hemolysis.